Hey guys, I'm Charlie Munford, president of the Agential Biology Institute. And in this lecture, I want to lay out the logical case for the scientific study of agency. So, this is the idea that organisms have a mechanism that operates at the level of the individual that allows them to make choices. So, words like agency, intention, and sentience can sound squishy and unscientific, even illogical. But I want to carefully show you that the hard logic that makes us sure that we need to look harder for a mechanism of biological agency before we can rest and say we have a good explanation for life. The theory we have was good, but new evidence has shown that it is a circular argument that amounts to claiming the genetic code changes the genetic code. So now we at ABI argue that if that argument won't stand up, we have to look for a mechanism that actually does the changing. The first thing I want to say is about the role of proof. In doing good science, we have to realize that no theory can logically be proven to be correct. This was a contribution of the philosopher Karl Popper in the 20th century. You can't look everywhere and measure everything that might pertain to any theory. So you have no certain logical proof that you didn't make a mistake. All you can do is make the best theory you can and see if we can find any evidence that disproves it. So no amount of supporting evidence will help if we find a solid example that contradicts a good scientific theory. If we find certain disproof, then we have to invent a whole new theory that explains and incorporates the new evidence. That's the only way to use logic in science. There's no way to use logic to establish a final theory beyond all doubt. So let's look at the theory of life and decide if that's the case now. The story of evolution that we have now, I will call the inheritance first view. So, inheritance first means that the causal responsibility for life lies in an inherited set of instructions that are edited by natural selection. That view has a very slippery trap in it that I want to lead you out of. So, most non experts have it in their minds that evolution says that organisms survive because of natural selection. So, this amounts to saying organisms survive because many close relatives of their ancestors failed to survive. Now, that's a non sequitur. The premises don't support the conclusion. The first step in understanding the situation is we have to catch that non sequitur and realize that selection is not a force that instructs organisms. The non sequitur isn't proof of a problem in the theory, it just doesn't prove the conclusion. It doesn't prove why organisms survive. This point reveals that the story of life is a story of variation. The important thing that is that selection doesn't figure into the story of the nature of variation. If you had no selection, life would still reproduce and vary, and you would still have evolution. Removing variance doesn't cause the variants that remain to then change in any specific way towards being better at surviving. So, variation is critical to the story. Now, if our story of variation is that there's no other mechanism for producing function other than natural selection, then the variants have to be blind with respect to function. They don't have to be random in every way, but they have to vary blindly with respect to whether they will have improvements to function. If they're not blind to function, then it means that information was already there before natural selection happened. So natural selection couldn't be the only source of instruction. Now, it's really hard not to fall into using this selection non sequitur. Even some very expert evolutionists make this mistake, and it's very difficult to hold the idea clearly in your mind at all times. What it means is we never go from selection to variation, only the other direction. So selection can't teach the organism how to vary itself. A variation begins with a parent, and selection allows that starting point to be some parents and not others. But it doesn't give a map for which direction to go in with your variation. Anything could vary, even things that should not vary, like essential steps of the Krebs cycle that run all the cell metabolism, or the few hundred essential cells in the brain that govern circadian rhythm, a hole in the heart, a hole in the skull, a hole in the spinal column. Even the delicate parts could change. That's what blind variation means. Selection picks parents, but it doesn't protect us from the emergence of any particular sort of new variation, even if it's bad. So let me repeat that in another way. If there was a change between the parents and the offspring, then selection had nothing to do with it. That's what blind variation means, and that's an essential feature of the inheritance first view. And it's very easy to slip into explaining changes by saying, well, selection prepared this organism to evolve in this direction, the direction that makes sense. But selection doesn't have a momentum, it doesn't have any foresight, and it can't give directions to the variations. So, therefore, if we see directions being given to variation, it shows us that there's a different mechanism at work than just selection. So this non sequitur, it comes up a lot when we look at variation. We have to use our imagination to picture what truly blind variation would be. It might just be a random hole in some structure or a tumor-like growth. When we see variations, for example, in the beak length of Darwin's finches, they often seem to be coherent, matched, smoothed variations. By this, I mean that the bottom beak is not only longer, but also the upper beak, and the tongue and the skull and the esophagus all fit together. The blood vessels and the nerves all match one another, and the nasal passages all fit together in the new beak. So, biology often puts variations together in a mysteriously clever way that seems to solve a whole class of problems at every turn. Now, the smartness of this is often attributed to prior selection that happened before the variation occurred. But as we just showed, this is the non sequitur. So we have to fight a suspicion that there may instead be some directional mechanism at work other than selection. So modern biology has erected a theory that has two main features to explain how life can be shown not to have that directional mechanism. One is a sort of lockbox, and the other is a code. So let me show you how these features work in the theory, and then I'll show you the new evidence that they're wrong. So logically, the number of offspring that can reproduce themselves has to equal one per parent per generation. Otherwise, the lineage will inexorably go extinct. So if variation was too wild, it would often happen that all the offspring in a generation would have some major defect. So you can imagine a fatal error in the cell membrane causing it to spill out its contents, or a cell becoming too salty or too hot or too acidic, or mitosis going wrong. So variation has to be confined within a small enough range that all the offspring don't by chance have fatal flaws. So this inheritance view is a story of a ratcheting mechanism, keeping the variation within tightly confined bounds so that at least one offspring every generation is functional. So this ratchet has to tighten and never loosen in order for life to become more complex. If the ratchet loosens, then the variants will likely all have fatal flaws. So this ratchet runs against the second law of thermodynamics, which suggests that disorganization and entropy should irreversibly hold sway over time. This ratchet prevents that from happening. So that's really interesting because when there are many offspring, like for example, a fungus producing millions of spores, there could be lots of wild variation and still you could make at least one good baby per generation. But if you're, say, an elephant and you have only two offspring per parent per generation, then you have to have a very, very tight ratchet to be sure that they're not at least two fatal errors in those offspring. So an elephant is a system of 4 quadrillion cells, so the complexity is enormous. So that's a really big challenge for the ratchet. There are an unbelievable number of ways that the system could go wrong, many, many degrees of freedom. You can imagine all kinds of mistakes. Maybe the aorta has a hole in it, or the esophagus dumps food into the lungs rather than the stomach, or the eyes get made out of bone cells rather than lenses, or whatever. And yet this elephant grows from one single tiny zygote that carries all the heritable information. So this situation presents a paradox. As the generations get smaller, the requirements for complex information get greater. So how is it that variation is so tightly constrained in an organism like an elephant? So to explain that, the first conceptual advance was made by August Weismann in the 1890s. And Weismann proposed that there were certain cells in complex organisms that carried their information behind a protective barrier so that all the variation that happens during life doesn't get in and loosen up the ratchet. He called that the germline. And that's the idea that the gametes are segregated behind a conceptual barrier that insulates them from variation. So this and Gregor Mendel's theory of particulate inheritance led to the modern synthesis. That's the idea that there's some molecular instructions in the gametes that control the development of the organism. So it's a sort of lockbox, a safe harbor against too much variation. That's the first main feature of the inheritance first theory of life. That's the Weismann barrier. And the second major advance in this explanation came from information theory. So Claude Shannon, working for Bell Labs in the 1940s, discovered that the information in a message could be quantified by specifying how many options there were for each position in the message. If you had a small number of options for each position, then a copy made of the message could be compared to the original and the errors could be corrected. And that was the insight that gave us the internet protocol and allowed basically the entire digital age for sending digital information. It was a big revolution to information. We could never use computers the way we do now, sending quintillions of bits of complex information all over the world, if we didn't have this digital error correction process. So in information theory, digitization led to error correction and led directly to this massive complexity. So that discovery in the 1940s gathered steam along with the discovery in 1953 of the DNA structure. So DNA turned out to be a digital code. Rather, it had only 4 nucleotides arranged into a very long sequence in a double helix. So nature had made the same discovery as Shannon, basically. When the DNA code was copied, it split into 2 strands, and the copy is compared to the original to produce a very accurate transmission mechanism. DNA polymerase makes 1 mistake per million base pairs or so, but with extra proofreading and repair mechanisms, the cells in humans make only about 1 mistake in 1 to 10 billion base pairs. So that's extraordinary accuracy. It's obvious that the information in the DNA is very important to life. And this cemented the importance of DNA in particular because only DNA, unlike RNA or anything else in the cell, goes through this painstaking error correction process because of its double helix structure. So that's the second main feature of the inheritance first story is the DNA code as the digital instructions of life. So that's really vital that only DNA is really important because of error correction. So in 1976, Richard Dawkins published a great book called The Selfish Gene. And it's a beautifully clear explanation that depicted all the work of living beings as the result of DNA sequences trying to get themselves copied. So many non-experts and even experts began to believe, in part because of Dawkins' book, that a selfish gene theory of blind variation was the only possible version of Darwinism, even though Darwin himself believed something very, very different. Charles Darwin actually believed variations were not blind, but were shaped by use and disuse, and that's what we now call Lamarckism. Richard Dawkins made many people believe that opposing the idea of blind variation was the same as arguing for God's existence, a claim that was very misleading. There's no reason why there can't be a physical explanation for Lamarckian variation that doesn't involve God, but that's not what Dawkins has argued for the past 50 years. He basically wanted everyone to see opposition to the selfish gene theory as opposition to science itself, and he largely succeeded. The theory the public now knows as Darwinism is not the Lamarckian theory that Charles Darwin believed, but a different version, and the version is most attributable to Dawkins himself. So all this reached a sort of genetic heyday in the 1990s, and the Human Genome Project was launched, and all the world expected that as soon as we knew in detail what the DNA code was, we'd be able to solve most or all medical problems by comparing the genetic instructions of one patient to another and working out why the system was misbehaving. So being able to read the instruction book of life should help you. Everyone expected that we would be going to the doctor with little copies of our DNA and thumb drives to solve our medical problems. And they assumed that there'd be a gay gene and a tall gene and a violence gene and a friendliness gene, all the traits that organisms have. So what went wrong? Why don't we use our DNA information at the doctor's office? It's very useful at a crime scene, just like fingerprints, but we also don't take our fingerprints to the doctor. Why did 23andMe recently declare bankruptcy after gathering the largest DNA database in the world? Why couldn't they use this information to make money from medical companies? To answer that mystery, we have to go back to the lockbox of the Weissman barrier and the complete code of digital instructions in the DNA. So both those ideas turn out to be wrong. So the first one is the lockbox, the Weissman barrier. That's the idea that the germline is segregated to protect it from errors. Well, so that one never held true in some animals, in all plants, in many fungi, in bacteria and archaea. So the vast majority of life. For one thing, most life on Earth is unicellular, and there can't be any germline if there's only one cell. So Weissman probably knew that very few branches on the tree of life actually segregated their gametes, but he had no idea how much complexity lower organisms could actually display. And we know now that even simple unicellular life forms have enormously complex behavior and many adaptations we still do not understand or have the ability to recreate. The lockbox and ratchet idea is that inheritance protects the information, so life should evolve like a family tree. But now we know that the tree of life isn't a tree, it's a web. It turns out that most prokaryotes have changed their DNA as much from borrowing or lending to totally unrelated lineages as they have by mutation of their own DNA. That's called horizontal gene transfer. This means that the information in the genome didn't get there by tightening the ratchet, but the opposite. It got there because bacteria under stress borrow and lend genes from each other promiscuously, even if they aren't related. They loosen the ratchets when times are tough. So this is causing a lot of antibiotic resistance in hospitals right now. Microorganisms cooperate together to an enormous degree, much more than we realized in the 20th century. In fact, our microbiome functions much like an organ would function, even though it's entirely made up of very small organisms with very different DNA from ours. And organisms have even fused together in symbiogenesis. For example, the mitochondria in our cells are descended from free living bacteria. In the 1950s, Barbara McClintock discovered that even in complex life like corn plants, when stressed out, organisms often change their DNA. So they do not create specific point mutations, but they do rearrange and duplicate their chromosomes. That's called transposition and duplication. In fact, this is now thought to be a main reason why new species are created, because a population becomes reproductively bifurcated when a new chromosome pattern emerges. Cancer cells do a lot of this, and often it causes cancer lines to become immortal, never aging. So today there are cells in labs all over the world used for research called HeLa cells. This line of cells came originally from a woman named Henrietta Lacks, who had a breast tumor that mutated into a particularly vigorous type. Often cancer cells develop enormous, weird, novel genomes through duplication and transposition. Far from being in a lockbox, it's turned out that the genome is quite often manipulated quite readily by the cell and by the conditions that prevail during the life of the organism. So when McClintock received the Nobel Prize for her work in 1984, she called the genome a sensitive organ of the cell. She understood that manipulation of the genome meant that organisms had some control over their DNA. Now, in complex metazoans like humans and mice, there are lots of other ways that germline cells are manipulated and changed by the organism during its lifetime. Some of these can be heritable changes. This is called epigenetic inheritance or RNA-based inheritance. For example, mice and humans have been shown to transmit small RNAs to their gametes that change traits in the offspring. This doesn't have to be inherited permanently to change the pathway of evolution. It can be applied again and again if it's a good adaptation, can even be selected for. That's called genetic assimilation. For example, mice have been exposed to electric shock after being given cherry blossom scent, and the sperm from these male mice was then used to impregnate females in the lab on a different continent who'd never smelled the cherry blossoms scent, and the baby mice, nevertheless, on this different continent, were born with a strong fear response to the cherry blossom scent. So there are many examples of soft epigenetic inheritance like this, including in humans. And it's very hard to argue that results like that come from blind variation. So now back to the second mainstay of the inheritance first view, the idea that all of the critical information is in the DNA code. So that idea has also been falsified. The first big surprise of the Human Genome Project was that complex organisms do not always have more DNA than simple ones. So humans have about 25,000 coding regions, which is far less than strawberries, for example. And there are amoebas that have 10 times more DNA than we do. That was the first surprise. The second surprise was that there isn't a direct link from specific sequences in the DNA to specific protein structures, let alone to body parts or traits like height or aggression or any complex diseases. So humans have 3 to 5 times more proteins than they do genes for proteins. And that's because proteins can be made in all sorts of alternate ways by the RNA that splices them. So many proteins, about half, are not even specific in their structure at all, but have large areas that are totally disordered. Now, remember, the theory requires that all the vital information about organizing the elephant lives in the sequences of the DNA. So, how can that information be preserved and expressed into an orderly elephant if there's so much disorder as it's decoded? Okay, another problem for the theory has been known for a long time. In Dawkins' version, the specific nucleotide sequences are supposed to give us the reason for all the attributes of the organism. Everything the organism does is in order to replicate those codes. But in sexual eukaryotes like humans, there's a step during reproduction called crossing over, where the sister chromatids swap sequences with one another. So this means that any given sequence stands a non-zero chance of being broken up during reproduction. Now that makes it logically impossible that this complex cellular behavior happens because of instructions in a code. Um, the behavior of crossing over can't be coded for if it scrambles the code that drives it, right? So you cannot have instructions which say scramble me every generation. If you did, the instructions would soon be scrambled and be lost. So it's also been found that the part of the zygote that has not been error corrected carries a lot of influence over development. For example, in an experiment that replaced the chromosome of a goldfish with those of a carp, the resulting offspring still had the vertebral structure resembling a goldfish. So that means that the supposed program for the vertebrae could not possibly have been in the genome. If it was in the cytoplasm of the egg cell, it means that the quote unquote program was not the sort of program that had been subject to the ratchet of natural selection that was supposed to be the only select explanation for life. Because outside the nucleus, there's no error correction procedure. So that's another loose ratchet. Okay. What is the big picture here? What is the point of all this? What conclusion do these problems with the theory point to? How else could a cell know how to make an elephant except by having inherited the knowledge from its DNA? Where else could all this information have come from? If DNA and natural selection don't tighten the ratchet, what does? So, unfortunately, this is a place where rationality has to take a back seat because logic can never show us that a theory is true. But and logic doesn't give us a new theory. Only imagination can do that. So, the only reasonable answer to what can tighten the ratchet, how that information can get into organisms, is the only reasonable answer is that organisms can learn, that they can learn that information. And that's what we call biological agency, right? So the only way that I can see rationally, logically, to solve the difficulty with the existing theory is to suppose, even though it seems crazy, that all organisms down to the simplest bacteria have some intelligence. They are aware to some tiny extent of the world around them, and they all, from the amoebas to aspen trees, have some ability to glean knowledge from the world around them. Inheritance happens, but inheritance has to be adjusted and improved by learning. So if this is true, then it logically follows that there must be some fundamental physical mechanism for learning that is not a result of natural selection. And in fact, if it's true, then the learning mechanism is probably the reason why we have individuals at all. It's probably that this mechanism of intelligence is creating the unit of selection that begins the whole story of life, the individual organism. It must be that all cells have a sensitivity to their world built in from the beginning. So, yeah, so the consequences of that are quite mind-boggling. It would mean that nothing we do is completely hardwired. Nothing is absolutely quote unquote natural at all. Everything is up for change to some tiny degree. It would also mean that It's the search for knowledge that drives life forward, not the instinct for survival. That's a big, big difference. Remember the non sequitur, we can't explain the thirst for new variations, new knowledge based on selection, based on survival and reproduction. So all the sociobiology that claims that our human nature is hardwired and our DNA hardwires us to do this and not that, all that has to be rethought. It also explains why variation has appeared to many brilliant biologists over many decades to be blind. Because if organisms have agency, then it would explain experiments where new mutations in bacteria are shown not to have reproductive advantages. Because biological agency isn't about finding reproductive success, it's about understanding the world. It happens at the level of the individual. What biological agency shows us is that in our cells, in our DNA, it's not a set of instructions at all. Our DNA is a set of memories. Everything we know because it happened to us and we remember it. And some fundamental things we remember from our distant ancestors in the form of nucleotide templates for making proteins. So when memories are in our minds, they instruct us, but they don't control us. And the same must hold true with ourselves. We can change our minds if we see a better explanation, a better way to live, and it looks like cells can change too. Survival through rounds of natural selection is very important, no doubt, and it explains a lot of evolutionary change. Logically, only surviving lineages are alive now, but that's a tautology. As far as we know, that's just the definition of being alive. We are descended from survivors. But that doesn't mean that everything we want to do has to be about survival. The world is more open to us than that. Anything that interests us, anything that gives us knowledge is open to us. And that same deep truth may be true for all living beings. They are all creative, thirsting for knowledge, willing to cooperate, willing to change if they see a better path, if they find meaning in a new way of being. I hope I've convinced you that the scientific search for the mechanism of agency is important and that it rests on solid logical ground, not just on squishy feelings. Of course, we all have strong intuitions about being free, about deciding for ourselves, about being sentient and aware and learning. But deep down in the hard science of evolutionary biology, there's also hope there in our most hard-nosed rationalistic theories for believing in progress, for believing in meaning and enlightenment for all life. Thank you.